Alright, we're recording. Hey AP Bio students. So I got another request to make another quick little video explaining a different topic of biology. Today we're going to be doing membranes and different cell transporters. I'm going to try to cover some of the material from your midterm study guide. I've highlighted the ones here. It's very selective. These were the topics that I still understood and still remembered over the years enough to make a video on it. So I hope this is helpful for those of you taking the class. Let me just move my desk to make it easier to record. Alright, so we're going to talk about membranes today. And just a disclaimer, this is going to be mostly from an anatomy physiology standpoint, just because from an anatomy perspective, just because that's what I've been trained in, rather than a cell biology perspective. But it should still be mostly similar. I would warn you that for biology, especially when it comes to transporters and cell membrane, the information, the way it's categorized, can differ a significant amount depending on which textbook you use. So the best resource to use in this case would be the textbook provided by your school in the AP Biology class. So I'll give you a general overview today, a general information so you can get yourself going. But to really understand the material to the specificity required for your AP class, you really do need to read the textbook and read the slideshow. Alright? Because every bio book is different, unfortunately. So there's no universal way to categorize things, at least among the different books. Alright, so we're talking about the cell membrane today. So the cell membrane is made of these things called phospholipids. Phospholipids, and already my marker is dying. It's made of phospholipids, and the phospholipid looks like this. It's got a head and a tail. So what happens is when you have a lot of phospholipids together in the cell, they form something that looks like this. I'm just going to draw a few of them. This is what makes up the cell membrane. Alright, this is called a phospholipid bilayer. A phospholipid bilayer. Alright, now an important distinction to make with phospholipids is that the head here, this is hydrophilic, which means it likes water. It likes water. The tail here is hydrophobic. And that means it hates water. So we know that the cell is filled with a fluid called cytosol, or sometimes it's called cytoplasm, which means that when you put phospholipids into a membrane, it orients like this, so that the heads are hydrophilic, they like water, they face the outside, so they're touching the cytoplasm, and the tails are hydrophobic, they hate water, so they're facing the inside, they're facing away from the water. So in the cell, this would all be water here on the outside, so the heads are facing the water because they like water, and the tails are facing inside because they don't like water. Alright? Now, an important thing about the polarities, because why do we care about these different layers? So in order to be water-loving, in order to like water, you have to be what we call polar. Polar means the charge isn't shared equally. And then likewise, if you hate water, that means you're going to be something called nonpolar. I'll write it in a different color. Nonpolar. So, if we look here, we see the heads like water, which means that the heads are polar. And this is polar. And then we look at the tails here, These are water hating, they're hydrophobic. So then this is, pardon my family in the background. There goes Dad again. Alright, these are hydrophobic. So that makes them nonpolar. Alright? So notice how we have polar parts of the cell membrane, and we also have nonpolar parts of the cell membrane. Which means that for a molecule to cross the cell membrane, since there's so much more nonpolar than there is polar, because look at it, you just have a thin layer of heads which are polar, the rest is nonpolar, which is so much bigger. Since the nonpolar region of the membrane is so much thicker and so much bigger than the polar part, in order for a molecule to cross the cell membrane, which means in order for a molecule to go all the way through to the other side, either this way or this way, it has to be nonpolar. Or it mostly has to be nonpolar because the nonpolar layer is so much thicker than the polar layer. Alright? So now, things that cross the membrane. I'm going to make a list for you. Things that can cross the membrane. Remember we said that in order for it to cross the membrane, it has to be nonpolar. So, some examples of that would be lipids. Remember, lipids are fats. Steroids. CO2 and O2. So lipids, steroids, carbon dioxide, and oxygen. All four of these things can just cross the membrane as it is. It can just move straight through. It moves in between these little phospholipids and it gets spit out onto the other side. Alright, these four things cross easily. There are also a few other things that can cross. We have ethanol, and we have water. Now, ethanol, this is going to be a little tricky because we haven't had organic chemistry yet. Ethanol looks like this. Alright, so we have a bunch of carbons, hydrogens, and an oxygen. And the abbreviated version is to draw it like this in organic chemistry. Now, water looks like this. You're going to have water, an oxygen, and two hydrogens. Whenever you have something, whenever you have an OH, an O bonding to an H in chemistry, like here, here, and here, and even here, that makes a molecule slightly polar. Alright? So ethanol and water are polar. They're actually polar molecules. But they can still cross the membrane because they are so small. So ethanol is pretty small compared to other biomolecules. Water is pretty small compared to other biomolecules, so they can sort of cross the membrane. But they cross, both of these things, they cross very slowly because they are slightly polar. So they will make it through a little bit if you give them enough time, but they won't go through nearly as fast as these other things that are non-polar, alright? So, those are the main things that cross the membrane freely. They just go straight through. So, something else is, well, you might ask, alright, well, this is non-polar things crossing the membrane. What about polar things? How do polar things get through the membrane? In order for polar things to go through the membrane, we have to have something in the membrane to help them cross. So here's the membrane. I'm just going to draw it like this. In order for most things to cross the membrane, you have to have what you call a protein. Alright. A protein helps carry polar things move through the membrane. So I'm going to say helps polar things cross. It helps them cross through the membrane, in or out. Now there's different types of proteins that help things move in and out, and that's what we're going to focus in on right now. All right? Alright, so what I'm going to do is I'm going to try to draw a little... I know it's a little tricky to keep track of all these proteins in your head, so I'm going to try to draw it in the graphic organizer. I'm going to try to visually draw it out. So we'll see how that goes. Alright, hopefully you can see the pen okay. So when we're talking about moving things across the membrane, we're talking about transport. Alright, let me see if we can focus. Oh boy. Alright. Sorry. It's technology getting in the way. Oh, I hate this thing. Oh, finally. Alright, so transport is things going through the membrane. There's two different types of energy, or two different types of transport. We have passive transport, and we have active transport. Alright, active and passive. Passive transport means that it requires no energy. Alright? Active transport means it does require energy. And the pen's dying. Why is everything dying today? Alright, so those are just the basic definitions that we're going to be working with today. Alright, so now we're going to look at the different types. Within passive transport, we have different types of transport that we're going to look at. Alright? So, what I'm going to do is I'm going to draw... it's going to look really crowded if I try to jam everything onto one page. So I'm going to split it up. So passive transport is going to go down here. I'm going to give passive transport its own separate page, okay? Just so it doesn't look as cluttered. Let me see if I can write it in marker. So this is all going to be passive transport. There's really 4 types of passive transport. Let's see if I can draw it. So one is going to be simple diffusion. Alright? Let's talk about simple diffusion. Simple diffusion is when something goes from high concentration to low concentration. So I'm going to bring in a sheet of scrap paper here. If we have a chamber with a membrane in it, and a lot of stuff on the other side. If I make the membrane permeable, simple diffusion just means stuff is going to move from high concentration to low concentration. So it just flows straight down the gradient. This is called a gradient, alright? So it's going to go from high concentration to low concentration. That's simple diffusion. The second type of passive transport is osmosis. So I'm going to write a little note. Simple diffusion is high to low concentration. The second one is osmosis. Osmosis has to do with water. And the thing you have to know about water is water flows, so H2O flows from high concentration to low concentration. And another way to say it is water flows towards salt. Alright? That's the way I learned in college. Water flows towards salt. And what happens is it goes through the membrane. Like we talked about here, water is one of those things that can cross the membrane slowly, even though it's polar, but it's small, so it can go through slowly. But it's not fast enough if we just let it go through. So if we just let it pass straight through, it doesn't go fast enough. So what happens is, in the cell membrane, we have these special channels that allow water to go through, and those channels are called aquaporons. Alright, these are special channels in the cell membrane that allow water to go through. Now, I talked to a professor once at UConn who still doesn't believe that aquaporins exist. So just know that for some people it's still theory that water goes through channels. Some people still think that water goes through the membrane, straight through the membrane without any help. But for the purpose of AP Bio, assume that water goes through aquaporins mostly. That's why I say biology, people still don't know the right answer sometimes. So you really have to rely on the textbook to get the test information. I'm going to skip number 3 for a second. We're going to go to number 4. Number 4 is called bulk filtration. Bulk filtration, this is not one you're going to see commonly in AP Bio, but bulk filtration means everything moves in the same direction. Everything in the same direction. Whether it's water, whether it's salt, whether it's protein, or whatever, it all goes in the same direction. An example would be in the kidneys. So, in the human kidney, it looks something like this. The kidney is made of these little repeating tubes called nephrons. This is called the glomerulus. It's a little tube that you have in the kidney. And next to that tube, you have the blood vessel. And it goes in and it barely scrapes it touches the tube. And what happens is, when blood goes through the blood vessel, when it comes to the intersection at the kidney, a big chunk of the blood just shoots straight into the kidney, into the glomerulus. And some of it goes past the other end. But a lot of the blood just shoots straight into the glomerulus, into the kidney. So all this blood that's rushing into the kidney, it doesn't matter if it's water in the blood, if it's cells in the blood, if it's proteins in the blood, everything is just moving in one direction. It's going straight down the kidney tube. So that's an example of bulk filtration. Alright? I'm not sure if they teach that in AP Bio anymore, but that, like I said, I'm teaching from an anatomy, physiology standpoint, because that's all I know. I haven't taken bio in a long, long time. Alright, so that's probably not a big emphasis on the exam then. Alright, now number 3. This is the big one. The third type of passive transport is going to be facilitative. Facilitative transport. Alright? And what facilitative transport is, stuff still goes from high to low, just like simple diffusion. So these two are very similar. It still goes from high to low. But what happens is, now we need to have a protein to help. So I'm going to say it goes from high to low, but there is a protein to help things cross. Alright, but there's a protein to help things cross. Alright, now we're going to look at the different types. And this is where, depending on the book, they might group things, categorize things differently. This is the way I learned it in college. within facilitative transport, within the proteins that help us move things, we can subdivide it into these things called channels. Alright? And a channel is something that looks like, let me just make sure I'm reading it correctly. Alright, a channel is something that looks like this. It's in the membrane, so here's the membrane. And when it's open on both sides, alright? It's open on the top and it's open on the bottom. But the key distinction with a channel is that when the channel is open, both sides are open. Alright? When we're talking about channels, what we're talking about a lot of times is an ion channel. And there's different types of ion channels, alright? And we're going to draw them out. We can have something that's called, I'm going to bring it over here, something called a leaky channel, a leaky ion channel. Alright? And a leaky channel is something that I'm going to draw here, back on our scrap sheet of paper. A leak channel is something that's really always open. So it's like this, always. And usually one type of leak channel only lets one type of ion through. So you can have one that lets sodium through. Sodium's going through, it just flows straight down. And then you can have a separate one for potassium. Potassium just goes straight through. Alright, so it's like, think of it as like different bathrooms. Just one type of ion going through. So that's the leaky channel. So normally these are open. Another type of channel you can have is called a ligand channel. Alright, and a ligand channel is still a channel, but the key distinction is that for a ligand channel, something must bind to the channel in order to open it up. Something binds to open it. So I'm going to draw here a ligand channel. So normally, if this is a membrane, the channel might look like this. See how it's not open yet, right? Maybe it's closed, it's not open. But then when you get something that comes in that binds to the channel, this is called a ligand. It's a signaling molecule. When the ligand binds to the ligand channel, after it binds, it's going to open up. So we opened it up. And now some of the ions, maybe it's sodium, maybe it's whatever molecule it is, can flow through. But it can only flow through when the ligand has binded to it. Once this ligand leaves, once this unbinds, then it's going to close again, just like what it looked like before. Alright? So it's ligand channels. Another type of channel is a mechanical channel. And a mechanical channel is something that is physically pulled open. It is closed until it is physically pulled open. So an example of that would be the human ear. So in the human ear, you have these are called stereocilia, but don't worry about that. And they have channels at the very top that look like this. So these are all channels. So normally they're closed here, but they're attached. This is a little door that's on the channel, but they're attached to a little string. When sound waves come into the ear, it causes these little cells to move, and when the cell is moved, what happens is it pulls on the string pulls on the door that's closing the channels, and it causes the channels to pop open. Alright? So now it looks like this. So we pulled on the string, we opened up the channels, I should make it open here, and now ions can flow through. Alright? So that's mechanical pull, when something pulls the channel open. The last type is called a voltage channel. This is very important. If you ever take any class in anatomy or physiology in college, the voltage channel is very important. Alright, because this is what runs our body. Because our body runs on electricity. So these are probably the most important types of channels in the human body. It's what your brain runs on, it's what your muscles run on, pretty much every function in the human body. There are diseases that affect these types of channels, and those people don't usually do too well, just because of how important they are. So these types of channels open in response to electrical change in the body. Alright? So what it looks like is, and this is kind of tricky to understand because we haven't taken physics yet, but a voltage-gated channel kind of looks like this. So it's closed normally. And voltage is a unit of measurement in electricity. Normally the cell membrane rests at about negative 71 millivolts. What is a millivolt? It's just a unit of electricity. And what happens is sometimes in the cell, the voltage, the electricity can change. So sometimes it jumps. The number jumps. It can jump from negative 71 to negative 55 millivolts. And when the channel detects that the cell has jumped from negative 71 to negative 55 millivolts, it opens the channel. Alright? So at negative 71, it's closed like this. But at negative 55, it opens like this. And then ions can flow through. Alright? And these proteins are all embedded in the membrane. And depending on which part of the body you're in, you might have more of one type of channel. So like your neurons, your brain has a lot of these voltage channels down here. Your kidneys have a lot of, we're not going to talk about kidneys. Your, what am I saying? Oh, the only type of channel that you might not have a lot of is the lead channels. You don't have too many of those in the body, but they are still there. Alright? So those are the channels. Another type of facilitated diffusion we're going to talk about is, sometimes these are called carriers or transporters. And these things are proteins that are open only on one side at a time. So in simple terms, it's like a revolving door. Alright, and I'm just going to draw it out. I'm going to give you an example too. So, there's a type of transporter in the, I believe it's the stomach, or maybe it's the small intestine, it's one or the other. It's called a GLUT1 transporter. It moves sugar across the membrane. So it looks like this. Now, what's the distinction between a carrier and a transporter, a carrier and a channel? Remember, a channel is open on both sides when it's open. A carrier is only open on one side. So what happens is, for the carriers or transporters, it looks like this, and then the sugar comes and it binds to it. So it's going to come in and bind to that site. When it does bind, alright, it is going to change shape. So it went from looking like this. The sugar has binded to it, and now it's going to change shape. So now maybe it looks like this. So now the sugar is inside the transporter. And then what happens is it's going to change shape again. And it's going to dump the sugar onto the other side of the molecule. So that's how it helps move sugar across the membrane. But do you see how even as it's being operated, the transporter is never open on both sides? It's only open on one side, and then it changes its shape and it dumps it on out the other end. All right, and then depending on the part of the body, it resets different ways. But you don't have to worry about that for AP Bio. All right, so those are the passive transporters. So what do all of these things have in common? They are all passive. So they do not require any energy to use them whatsoever. All right? Now, remember what we said? That was going to be... So when we talked about our entire thing of transport, that was passive. Passive was all of these things. Now we're going to talk about active on the other side. All right? Active, I'm going to draw an arrow so we can expand it onto a new sheet of paper. So remember, active requires energy. Maybe I'll write in red just so we know. So active, it requires energy, like we said here, and a lot of times it goes from low concentration to high concentration. So normally we go from high to low, which was passive. But to go from low to high, now you're going to need some energy. Alright? So, there are a couple different types. Actually, it's really only 2 types that you need for AP Bio. Active transport. You can have primary active transport. Primary active transport, like we said before, it goes from low to high concentration, but the key distinction with this is it directly uses ATP. It directly uses it. So we're using ATP. Now what is ATP? It's something, adenine, adenosine, whatever it is, triphosphate. Alright? And an example of this is the sodium-potassium pump. I'm going to draw it on another sheet of paper. You probably talked about this in AP Bio. Now, I'm going to be honest, I didn't really understand what the purpose of this was until I took anatomy physiology in college. Fun fact, I actually skipped biology in college. I never took biology ever. So a lot of times the AP Bio curriculum I look at and I'm like, whoa, I haven't done that since 10th grade. Let's see. So the sodium potassium pump. I'm going to bring up my old notes here. You can tell biology is not my favorite subject. It's probably my least favorite subject, now I think of it. Chemistry is also my least favorite. Pretty much all science. So, what you have is, the goal of the sodium potassium pump is that it pumps Every time it pumps, it's going to pump 3 sodiums out, and it's going to pump 3 potassiums in. Alright, so sodium gets pumped out and potassium gets pumped in. And a neat little memory trick that I read about to remember this is that the sodium-potassium pump, if you remember the word pumpkin, like Halloween, is that it pumps K in. It pumps potassium in. Alright? That's just a neat little way I remember it. And the way it does that is, it looks like this. So we have a cell. Now, cells, this is going to be the outside of the cell. This is going to be the inside. Alright, so on the outside of the cell, this is pretty much most cells in the body have high concentrations of sodium. The inside of the cell has high concentrations of potassium. Alright? So, what happens is, the pump works such that when it's open, when it's facing the inside of the cell like this, 3 molecules of sodium bind to the inside of it. Alright? And then we add a phosphate from ATP. Once that phosphate gets added, the membrane is going to flip its shape, or the protein is going to flip its shape, and it's going to kick those 3 sodiums out. Alright? It's going to kick those 3 sodiums out. So notice how sodium was high on the outside, and it's low on the inside. So the inside is low sodium. But since we're moving sodium from low concentration to high concentration, that's requiring energy. That's what makes it active transport. Alright? Now, what happens is, phosphate's still there the whole time. Now we're going to get some potassiums that are going to come in. When the membrane is facing this way, 2 potassiums can come and bind to it. Alright? And then the phosphate breaks away. It releases itself. It just goes away. And then the protein flips its shape upside down again. And then the potassiums get released into the cell. Alright? So, once again, we look back at the cell. Remember, the outside of the cell has high sodium and low potassium. The inside has low sodium and high potassium. Since we're moving potassium from low concentration to high concentration, that requires energy. So that's why we have this pump. Now, something that I want to mention, because this was on the study guide, is notice, I'm going to draw just a cell here. Alright, so every time the sodium-potassium pump runs, we're kicking out 3 sodiums. Sodium is positive. And we're bringing in 2 potassiums. Alright? But now all of these ions are positive. But do you notice how we're kicking out 3 positive charges and only bringing in 2 positive charges in place? That means that we're losing, if we just go by logic, if we're losing 3 positives and only getting back 2 positives, we're losing a positive charge, right? So every time this pump works, every time it runs, we lose 1 positive charge from the cell. Now what happens is the negative charges, they're not moving. They're not moving in the cell, alright? Because the pump only moves sodium and potassium. So the negative charges aren't changing. They're just staying in the cell. Fun fact, the negative charges, do you know what they are? They're mostly DNA. DNA is the main negative charge inside the cell. But anyway, so we're moving positive charges without touching the negative charges, and since every time the pump runs, we're losing 3 sodiums and only gaining back 2 positive potassiums, that means that every time we lose 1 positive charge and the cell becomes more negative. The sodium-potassium pump makes it more negative because we're taking out 1 positive charge each time without ever replacing it back. So that means that electrically, the cell is negative. So the inside is more negative. And when we measure it using electrical units, we get that the inside of the cell is negative 71 millivolts. Alright? That's where, when we talked about earlier, the voltage channels, that's where this negative 71 comes from. Alright? It's the negative charge. The cell is made negative by the sodium-potassium pump. Alright? It's at negative 71 normally. And because of that, because the sodium-potassium pump made the cell more negative, we say that it's polarized, the cell. And what polarized means is that there's a different charge on the inside and outside, alright? So difference in charge between the inside of the cell and the outside of the cell. So that's how this protein helps to polarize the cell. It establishes a charge. Alright, the other type, the last type of active transport is going to be secondary active transport. And what this does is, the definition is, it does not use ATP directly. So it uses no direct energy usage. Alright. Now, in college, there are kids that finish general anatomy physiology who still don't understand how this works. So, I'm not going to go too in-depth, because I don't want to confuse anybody. But what happens is, when you have secondary active transport, you're going to have 2 molecules. There's 2 molecules involved. One molecule goes from high to low, and the other molecule goes from low to high. Alright? So, I'm going to give an example here. And don't worry, don't freak out if this doesn't totally make sense at first. Alright, this is going to be the sodium and glucose exchanger. This is actually how our sugar is absorbed in the body through the small intestine. Alright? And I'm just going to draw it. So we have a membrane here. So on the outside of the cell, we have high sodium and low glucose. On the inside of the cell, we have high glucose and low sodium. Alright? This is the outside, this is the inside. Alright, this is where it gets a little tricky for people, so I'm going to simplify it. So sodium, remember what we said with secondary active transport, we move one molecule from high to low. Alright? In this case, that's going to be sodium. I'll just write it. For our example, it's going to be sodium, but this is... I'm not going to write it here because it's not always sodium, but in this case, it's going to be sodium. So we have sodium. It's going to move into the cell. Remember, sodium is going from high to low. So that's normal. That's the first one. as sodium is going from high to low, it's going to generate a force that's going to push glucose into the cell along with it. But do you see how sodium is going from high to low? Glucose is going from low to high, because it's going from the outside to the inside. It's going from low concentration to high concentration. But because we're pairing it with sodium going into the cell, that's what allows glucose to go from low to high. So notice that we're not using ATP. There's no ATP here. There's no ATP. There's no direct energy usage. But the fact that we're using sodium going from high concentration to low concentration is what generates the force to push glucose from low concentration to high concentration in a direction that it wouldn't normally move. So that's secondary active transport. You're going to couple one thing that's moving from high to low, in this case it's sodium, and you're going to couple it with something going from low to high, which is glucose. Alright? And this is the reason why, this is how sodium and sugar moves into our small intestine, into our bloodstream. So that's why it's important to have both salt and sugar in your diet, because it helps both of them move in. Alright? And just to expand on that idea, for secondary active transport, I don't think you'll need to know this for AP Bio, but just in case, you can have something called a symporter. And that's when two things move in the same direction. Just like our sodium and glucose. So I'll put that there. Sodium and glucose. Across the membrane. And then you can have something called an antiporter, where two things move in opposite directions. Alright? And I can't remember an example off the top of my head, but there is one I remember. I just can't remember the name. Alright, so that's cell transport. Hopefully that made a little sense. Now, I did want to talk a little bit about water. Osmolarity. Just quickly before we leave. So water, as we can see, was, think to yourself, is it active or is it passive? So hopefully you're thinking passive, alright, because it was on our blue flowchart. Osmosis. We're going to talk a little bit about this. So water goes from high concentration to low concentration. And there's different types of solutions. You can have a hypotonic solution, and you can have a hypertonic solution. And when we're using hypotonic, hypertonic, they're comparative terms. But in general, a lot of times we're talking about the solute, the fluid that we're going to put things in. So we're going to put a cell into water, into a type of water. Hypotonic means the solution has less salt. Hypertonic means it has more salt. And this is all relative to the cell. So here's your cell. A hypotonic solution doesn't have a lot of salt. Alright. Whereas a hypertonic solution has a lot of salt. I'm just going to draw it like this. So when you're doing these kinds of problems, the best way I can think of to do it is you're going to be given maybe a percentage or molarity amount, concentration amount, but the way I like to do these problems is when they're asking you which way is water going to flow, I always label each side. So this is the outside of the cell. We have a hypotonic solution, which means the outside has less salt, so low salt. When we have low salt, that means we have high water. Now let's look at the inside of the cell. The inside of the cell, remember, since the outside is hypotonic, that means there's less salt outside than there is inside. So in the inside, that means there's going to be higher salt. And when there's high salt, there's low water. Alright? So now, after I label both sides, the outside and the inside, I forget about the salt, and I just focus on the water. Water goes from high concentration to low concentration, so I draw an arrow from high to low. Because the outside is high, the inside is low, we're going from high to low. So in this case, the water moves into the cell. Alright, let's do the same on this side. So, hypertonic, that means the outside has more salt. So the outside is high salt. When we have high salt, we have low water. Now let's go to the inside of the cell. There's more salt on the outside, because hypertonic means the outside solution is more salty. So the inside of the cell is low salt and high water, because the two are always opposites. So now, I forget about the salt, and I just look at the water. Water goes from high to low. We go from high to low, so water goes from the inside to the outside. So in this case, water moves out of the cell. So that's how you solve these problems. Alright, so we talked about water, we talked about transporters. One of the other things, quickly, I want to show you before we leave is how to study all this information. So I know for biology, a lot of times it's kind of hard to keep all the information straight. The biggest piece of advice I can give you for any bio class ever is you have to read the textbook. So the textbook, let's make sure the recording's okay. Looks like this is my textbook from college. This is what I had for general anatomy physiology. You have to read the textbook. And you can see this with like kids in college. Those that read always tend to score higher than those that don't. And what happens is, I'm just gonna go to the section as an example on transporters or transport, because that's what we're talking about today. You have to read the book. And let's say you cover, let's say, a chapter in class, and it's like 20, 30 pages in the textbook. That's 20, 30 pages you have to read in order to understand it well. And I would not recommend using Google, because, as in biology, I've tried looking at it in college. Google gets it wrong half the time, because depending on the book, they can categorize information differently. So, one of the things I struggled with in bio was how to read a textbook. And let me show you how to take notes from a book. The thing that you don't want to do is you don't want to go sentence by sentence. So what you want to do is you want to go read one paragraph at a time, alright, before you write something down on your page. So I'm just going to give you an example. Let's look at, can you even see this? Move down. Try to focus it again. Alright, so let's look at this paragraph on active and passive transport. So, what I used to do when I first started was I always used to read a sentence and then write a sentence in my notebook, and then read a sentence and then write a sentence. What you want to do in bio is you want to look at the entire paragraph first. Read the whole paragraph before you write something down in your notebook, into your notes. So I'm going to read it. Active transport can be divided into two types. Primary active transport, the energy pushes molecules against the concentration, comes directly from ATP. Secondary active transport uses potential energy, stored in the concentration gradient of one molecule, to push other molecules against the gradient. Secondary transport ultimately depends on primary transport because the concentration gradient that drives the secondary transport is first created using energy from ATP. Alright, so I read the paragraph. Now I have to understand the paragraph. And what happens is I have to be able to say in my head what it means. So, what it's saying is that primary active transport, primary uses energy. Secondary doesn't. It uses concentration. And the concentration itself comes from using ATP. So that's a little confusing, just because I didn't explain it as in-depth as this book goes into. But I'm just showing you for the purposes of note-taking. Now that I've summarized it in my head, I'm going to write it in my notebook without looking at the original book. So when I'm writing notes in my book, I should not be looking at the textbook. Because the purpose is you want to take this textbook and you want to put it in your own words. Because when you translate it into your own words, that's what's going to help it stick in your head. So when I take notes, I'm going to say primary transport, it uses energy, uses ATP. Secondary, it uses no ATP, but it uses concentration gradient. So do you see how I condensed that entire paragraph into just 2 or maybe 3 bullet points? And you notice how I didn't really have to add all that extra detail they gave in the last few sentences. So you would do that for this page and the next page, and the next page, all these different pages until you reach the end of the chapter. And then what you should have is a translated version in your own words that is super, super short. Alright? And the idea is since you're putting in your own words, you're already starting to memorize it. So that's how you take notes from a bio book, from my perspective. And then once you've done your notes in your notebook, when you go back to study, you're going to take the notes that you take, and you're going to turn it into these neat pictures, just like me. So the best way to study your notes is to draw them out, alright? Make drawings, make things into charts, kind of like what I did with all these different channel types. Just have a little fun with it, so that you're able to make sense and organize all the information. So I would highly recommend drawing charts rather than just rereading your notes. Alright, so I'm going to go very quickly through some of these questions on the study guide. I'm going to go fast, but it's because we covered a lot already. So how does the structure of the phospholipid maintain the internal environment of the cell? So the phospholipid bilayer is talking about the membrane. What happens is it controls what enters and what exits. Alright? Now, this is clearly not as much detail as you should put in when you fill out the study guide, but this is just a rough introduction to it. So when you're studying, go back and fill in more. What role do phospholipids have in maintaining the internal... that sounds kind of the same as this one. What role do proteins have in maintaining the internal environment? Proteins control the movement of polar substances. What is the structural framework of the fluid mosaic model? Fluid mosaic is a fancy word for this picture that I drew out. The membrane is made of phospholipids. The cell membrane is a phospholipid bilayer with proteins added in, with proteins embedded. How does this cell structure... What does the structure of the cell membrane result in the selective permeability? How does it? The answer is there's proteins that control what crosses and what doesn't. How does the membrane separate internal from the external? It's a barrier. What is the direct consequence of membrane structure? Who made these questions? I don't know what she's asking about here. What's the direct consequence? I'm assuming it's just... It's that not everything can cross. Because remember we said non-polar things cross easier than polar. What types of molecules pass freely across the membrane? Oh, that's an easy one. Non-polar. And if we remember from our list, it's lipids, steroids, carbon dioxide, oxygen, and a little bit of ethanol and water. What types of substances move across channels? That's going to be polar things. What can pass through the membrane in small amounts? Let's say water and ethanol. Ethanol is a type of alcohol. I'm going to skip these because I really have no idea what fungi, prokaryotes, cell walls are, what they have. How does passive transport move things across the membrane? It moves through either diffusion or through channels, transporters, or carriers. How does active transport move things? It pumps using ATP, just like that sodium-potassium pump we saw. How do concentration gradients form across the membrane? That's a tricky question. How do gradients form? The answer to that is actually pumps. Like the sodium-potassium pump. And the reason being is that, remember when we were talking about how the pump pumps sodium and potassium against the direction they want to flow in? What happens is that ends up separating the charges. So you're pushing more of one ion out than bringing it in. So it makes it so that there's more sodium on the outside and less on the inside, and there's more potassium on the outside and more potassium on the inside. It's not as important in this class, but when you take anatomy physiology, this is super important. How do large molecules move into the cell? We didn't really talk about it today, but the answer is endocytosis and exocytosis. Endocytosis means you bring it in. Exocytosis means you bring it out. And that's pretty much all I know about that topic. I never took a cell biology class, so sorry if some of my explanations are a little vague. Why are proteins required for diffusion? Because polar things cannot cross. How do large quantities of water move across? The answer was aquaporins. Remember, some water slips through the cell membrane, but most has to go through the channels, aquaporins. How do membranes become polarized? The answer is, at least what I think she's trying to get at, is the sodium-potassium pump. Alright? And it has to do with this diagram we had right here. How we're pumping out 3 positives and only bringing in 2 negatives. So since we're losing 1 positive charge, the cell becomes negative. What molecules are necessary for active transport? Active. So ATP tends to be the big one. When is ATP needed for transport across the membrane? This is going to be primary active transport. Alright, and an example would be the sodium-potassium pump. How does the sodium-potassium pump, whenever you see ATPase, that really means, that's a fancy way of saying a pump that uses ATP. How does it contribute to membrane potential? I wish you were here the day I gave the guest lecture in Mrs. Babiak's AP Bio class, because during that day I gave a whole talk on what membrane potential is. Membrane potential is a fancy way of saying the difference in charges. The difference in charge between the inside and the outside of the cell. So that's what we talked about here. The inside of the cell is more negative compared to the outside because the sodium-potassium pump is pumping out 3 positives and bringing in only 2 negatives to replace them. So the inside of the cell becomes more negative, and since the inside is more negative and the outside is more positive, or it's less negative, that's a difference in charge, which means that we have a membrane potential. Membrane potential is the difference in charge across the membrane, or the difference between inside and outside. So, how does it contribute? It's because it doesn't pump equally. What are different ways to describe tonicity? We have hyper and hypo. And I forgot to mention one type. It's called isotonic. Isotonic just means equal. How do differences in osmolarity impact the movement across the water? It either moves in or out. Alright? And just go back to that diagram we drew. How does constant movement of molecules across... I do not know this one. Basically, it's not too good to have too much of one molecule on one side of the cell. So we need to move them across the cell to balance them out. And in reality, this is very important when you take more advanced bio classes like neurobiology or anatomy physiology. The way that your body runs is electricity. And the way that electricity is generated in the body is by moving things across the membrane. But that's more advanced than we have to go into here. So we covered quite a bit in this long lecture today. I know it was supposed to be fast, but I guess it wasn't really. I hope this helped. Let me know if you have any questions, and best of luck with AP Bio. See you next time.